everybody, I'm Louisa Diva and welcome to today's photosynthesis webinar. Today I'm very excited to introduce to you our speakers, Professor Thomas Nagele from the Ludwig Maximus University in Munich and Professor Erik Murchie from the University of Nottingham. These webinars are brought to you by the Capitalise programme, which is an Horizon 2020 initiative improving the efficiency of photosynthesis by exploring natural variation in core elements of photosynthesis. The aim of the webinars is to promote collaboration and communication amongst the photosynthesis community. So I encourage you to ask questions at the end of the talks, and you can do this by typing into the question box. Without further ado, I will introduce you to our first speaker. Thomas Nagele is a professor in plant science at LMU, and his work focuses on quantitative analysis of plant-environmental interactions. His lab applies experimental techniques such as pulse amplitude modulation, gas exchange, photometry, chromatography, and mass spec to resolve dynamics of plant metabolism in dynamic environments. He's established a platform for experimental high-throughput analysis of primary and secondary metabolism on subcellular scale, and his team develops mathematical models to simulate complex plant environmental scenarios. And today he's going to talk to us about stabilizing photosynthesis in a dynamic environment and asks, is it a regulatory trade-off? Over to you, Thomas.
Thank you very much, Louisa. And thanks, of course, for having me here and for inviting me. I'm really happy to be able to give this talk here. So as already introduced, my talk is entitled Stabilizing Photosynthesis in a Dynamic Environment. And of course, this is nothing new in principle. However, we were asking the question, is there a certain regulatory trade-off which we have to consider, which might be even hidden up to now? And why did we get this idea? So let me shortly introduce you to this idea. So we all know that photosynthesis is a quite complex process comprising several aspects, which we typically term into primary reactions, secondary reactions, and they perform capturing, interconverting light energy into chemical energy. And this is, of course, done by the interconnection of electron transport chain buildup of a proton gradient, which is then applicable to the Kelvin cycle, which can be subdivided into different substages of carbon fixation, reduction, and regeneration. And this, of course, this interplay matters a lot, and we see this especially if we perturb this system, for example, by changing the temperature regime or shifting the ratio of CO2 to molecular oxygen, because both are substrate for Rubisco. And due to this, due to the dual function, catalytic function, we also have to consider what happens to both products, 3-PGA and 2-phosphoglycolate afterwards. So I will come back to this in a second. And what we do in our lab is we ask the question, how can we mathematically put these processes into numbers in order to come up with a representative simulation of these systems, which give us some further idea of what is going on in the background of those very simplified pictures here. And then we typically perturb this system in order to see what happens to the system, how we can perhaps also modify our models, adapt them in order to get better and a better picture of the truth. So what we know is, for example, for shifting temperature regimes, is that there are immediate consequences, physical constraints, maybe due to thermodynamics, which reduce, in the case of a cold shift, immediately reduce Brownian motion, which has an immediate impact on reaction velocity, membrane, leads to membrane rigidification, and so on. And if a plant wants to grow or aims to grow in such a changing environmental regime, it should somehow at some point be able to adequately react, acclimate, or even adapt to this changing environmental condition. So a typical conserved and of course simplified picture which arises and which we know based on lots of great biochemical and physiological studies is that typically temperature induces a change, a dynamic change of calcium fluxes, which finally end up in a signaling cascade, which modify or even change signaling cascades and enzyme activities, which finally, if the plant successfully adapts or acclimates to this temperature regime, leads to stabilization of photosynthesis, and this then leads to stabilization of carbohydrate metabolism. So carbohydrates are, of course, direct products of photosynthetic CO2 fixation, and there's, as expected, a strong interplay, regulatory interplay. And there are lots of cascades involved. There are lots of molecular switches involved, and some of them are well known and described, and some of them are maybe a bit tricky to unravel and to study due to low abundance or due to complex interplay in a complex metabolic network. I just want to mention some of the very most prominent examples here. So what we know is that, for example, non-photochemical quenching, but also regulation of enzyme activities in the Calvin-Benson-Bessem cycle must be fine-tuned or tuned at all in order to stabilize this interaction between photosynthetic CO2 uptake and carbohydrate metabolism and metabolism in general. While under heat, what we know is, for example, that there's process of photorespiration being induced, and this contributes to stabilization together with the regulation of the Calvin-Benson-Bessem cycle. So, and with this, here in this talk today, I would like to focus a little bit here on photorespiration and its regulation in a in terms of system dynamics. So what is photorespiration all about? So photorespiration is the, at least that's what we usually tell students in our lectures, is the process which recovers typically 75% of carbon, which is lost. We now say lost, yeah. Typically, initially we say we term it to lose carbon in this process. When Rubisco catalyzes the oxygenation of ribulose 1,5-bisphosphate. And what we get is a molecule of 2-PG and a molecule of 3-PGA. 3-PGA can be used then, for example, to build up starch, to build up soluble sugars, and so on. While 2-PG undergoes then a complex multi-compartmental interconversion process and pathway, which is termed photorespiration. And there are lots of questions related to this pathway, and lots of great studies have shown what this pathway might comprise and affect. I just give you some ideas here. So it affects, and it is heavily involved, of course, in the carbon-nitrogen interface. So it plays a role in nitrogen assimilation, mitochondrial respiration. We have learned that there's an interconnection to the TCA cycle in the mitochondria, of course. It is contributing to C1 metabolism by providing activated intermediates, but it also touches sulfur metabolism, for example. And the physiology behind this pathway is, and I just named some of the important functions here to prevent, it is sought to prevent and reduce photoinhibition, to balance ADP, ATP, and NADP or NAD to NADPH, NADH ratios, but also to build up and biosynthesize amino acids, for example, glycine and serine, and to serve as a source of activated compounds. However, there is an obvious trade-off between all those interactions, because the yield loss shown here as an example for C3 plants might rise up to 50%. This heavily depends on the environmental conditions, on temperature, on the ratio of CO2 to O2, and so on and so forth, of course. Further, in recent and previous studies, and I just want to cite one here, which showed that there's, of course, lots of regulation going on. So biochemical regulation, molecular regulation. And what has been shown, for example, is that the intermediate 2-phosphoglycolates, so 2-PG feedback inhibits enzymes of the Calvin-Benson-Bessem cycle, and very prominent examples are here triose phosphate isomerase and cerebrotulose 1,5,1,7-bisphosphatase, abbreviated TPI and SBPAs. And this, of course, then has an immediate effect on the regulation of metabolism, leads to an alteration, can lead to an alteration of carbon allocation between CBBC and starch metabolism, but also, of course, as a consequence of that, to soluble sugar metabolism. Now, what interests us in this context is how can we use this information in order to get some more or some better understanding of the system responses, the system dynamics, which result from such feedback inhibition, for example, of 2-PG on TPI or SBPAs. So we, in our study, or in our studies, we follow or we aim to resolve system dynamic consequences coming from biochemical evidence. And what does it mean? It sounds quite abstract. I show you here in a simple example. If you have 3 cellular components, which might be metabolites, yeah, A, B, and C, and they are interconnected enzymatically, and you perturb this little system here, you can interpret this first arrow, for example, as photosynthesis, and the second arrow as mitochondrial respiration, entering and leaving the system. If you perturb the system and then observe how the system reacts, then you might observe different patterns depending on the structure of this metabolic network, but of course also on the underlying kinetics. Yeah, and what you typically see is, and what is well known also from other pathways, glycolysis is a famous example, is that metabolic feedback inhibition, as indicated here by those red arrows, stabilizes system behavior after perturbation. However, so that's maybe also well-known phenomenon. However, it now also depends on where this feedback inhibition emerges from. What we and others, lots of other studies, of course, coming from system theory have shown is that, for example, here the feedback inhibition coming from A on the reaction which interconverts C to A is more efficient in terms of stabilizing the system behavior than the feedback inhibition from C on the interconversion from B. Why? Because it directly gives a feedback from the input function here. Yeah? So that's a quite simple, however not trivial finding here, which we can fully understand. However, of course, we want to apply this principle now to larger networks, for example, to the network of the CBBC and of the photorespiratory multi-compartmental pathway. And how do we do this? Let me shortly give you a brief introduction to what we did in order to explain and also clearly define our findings here. So what we did is we created a model based on ordinary differential equations, an ODE model, which describes changes in metabolite concentrations over time as a metabolic function. And this metabolic function F depends on metabolite concentrations in the networks, enzyme parameters, and time. And then what you typically do is, in order to simplify this complex mathematical problem which emerges from a large network, is you linearize and you transform the your formula a little bit in order to make it applicable to and also to interpret, to make interpretation a bit more easy. Yeah, let's put it this way. So what we do is we, instead of focusing on the sole changes of metabolite concentrations, we are considering fluctuation terms. This roof on this metabolite matrix here, S, or vector S, describes the fact that, or describes the state after perturbation. Yeah? or the difference, let's say, between the metabolite concentration and the mean value of all of the metabolite concentrations. And if you go a bit more into mathematics, and I will just show you the results here of this, is that then you will see that you can describe those fluctuation terms by an exponential function. And this exponential function then is related to a matrix J, which is called the Jacobian matrix, and you can then later term those as so-called eigenvalues. Those eigenvalues give you already an idea about the solution of your system, and also, and we will see this in a second, also how this system might behave if you push it a little bit by, for example, transferring plants to low CO2 or high temperature or low temperature. So in order to show what this means in a less abstract manner, what you see here is a simulation over time of two metabolic states termed X and Y, any arbitrary metabolites behind that. Yeah. So we've got one input function which feeds X, and then X is interconverted into Y, which leaves the system. And then you can use, what we do is we fit our model to experimental data, then we simulate it, and then we introduce a perturbation, which you see here. So X or Y is perturbed, and then we take a look at the system behavior. And what you immediately see here in this example is that obviously this system is successful in coming back to the original level, so the original homeostasis, and that's why we term it to be stable. We can characterize this by negative eigenvalues because those negative eigenvalues lead to a decline of this exponential function, which indicates asymptotic value. On the other hand, if you have eigenvalues with a real part in it, which is positive, then you typically observe a system behavior like that. Yeah? So there's no coming back to the original homeostasis, and that's why we term the system to be unstable. You might already see it here. So this RE abbreviation indicates that the eigenvalues are complex numbers. So they consist of a real part and an imaginary part. We will focus here solely on the real part, because if we find that they are negative, then we say that the system behaves stable. If it's positive, we term it unstable. And what we did was we now generated a mathematical model of the CBBC together with the photorespiratory pathway, simplified it a little bit to focus on the central carbon interconversions. And what you see here in black arrows on the left is the CBBC with an input function and export functions towards starch biosynthesis and sucrose biosynthesis to the cytosol. And in orange, you see the photorespiratory pathway attached via peroxisome mitochondria going back to the peroxisome and coming back to the chloroplast. Then we put all this into a so-called structural kinetic model, which allows us to estimate the Jacobian matrix. And this is key to this approach. Yeah? So we apply a little trick here. We simplify and parameterize, we parameterize and normalize these approaches by a steady state value, which makes our life much easier. I won't go into too much detail here. However, you might believe me that afterwards you are able to estimate how this metabolic system behaves if you perturb it. Yeah? So this is directly connected to experimental data, to experimental evidence, which we use in order to find feasible solutions. As soon as we have such a feasible solution, we can perturb the system, and then by the entries of this Jacobian matrix and its eigenvalues, estimate how the system would behave if it is perturbed in a certain direction. And that's exactly what we did. And the first question which we asked was, is photorespiration necessary in order to stabilize the Calvin-Benson-Bessem cycle? So this question originated from the fact that you can also write the black arrows on its own and the orange arrows on its own. And then you might ask, how are they interconnected and why are they interconnected, right? And what we found was that if you cut off these orange arrows in our model, this leads to a destabilization of our Kelvin-Benson-Bessem cycle. You see it here that our solutions, which comprised several hundred thousands of simulations of different parameter constellations, so quite representative and statistically significant, this reduces the solution, so the number of stable solutions for the Kelvin-Benson-Bessem cycle compared to the full model. So full model stands for the textbook model, which we think is true, yeah, for comprising both CBBC and photorespiration. And further, we then ask, how specific is this contribution of the full photorespiratory pathway? So might it also be possible, or even better in terms of stability, to not go through peroxisome mitochondria and come back to the chloroplast? And that's why we inserted here in our in silico model an unspecific 2PG efflux, to get rid of this inhibitory compound. And what we saw was that the stability statistically increased. What does that mean? So for us, there were two conclusions from this. We concluded that photorespiratory pathway stabilizes the Kelvin-Benson-Bessem cycle, and we found by correlation analysis that glycylaldehyde 3-phosphate has a very strong effect on this stability. And second was that an unspecific efflux of 2PG with no recycling, not coming back to the chloroplast again, so we lose carbon here, is even better stabilizing. And this brought us then to the idea, okay, this seems to be a trade-off function of photorespiration, right? So you stabilize the system, however, you have also to account for a little bit of carbon loss. It is, however, better than fully stabilizing the system and losing all the carbon due to oxygenation. Then the second hypothesis which we tested with our model is, how does the efflux of carbon in direction of sucrose biosynthesis via the TPT transporter stabilize? Does it stabilize and matter at all? And what we found was that without photorespiration, you need much more sucrose, or you need to shift the carbon flux into sucrose much more than with photorespiration in order to come up with a similar stable solution. What does that mean? For us, it means that photorespiration decreases, on the other hand, the stabilizing effect or necessity of assimilate export, right? And this might hint towards a function, a physiological function. In other words, you need photorespiration in order to be able to decrease the ratio of sucrose biosynthesis compared to starch biosynthesis. In order to stabilize metabolism, but still biosynthesize enough starch to survive the night, you should have photorespiration, because otherwise, without photorespiration, you would have to shift the carbon allocation much more towards sucrose biosynthesis in order to come up with a stable network and with a stable kinetic system. And further, in the third scenario, we also tested a possible contribution of secondary metabolism. Secondary metabolites are typically induced under stress. So the idea was, okay, how does this all work together? And what you see here is comparing the full model and the model which lacks photorespiration. So, and a general rule, the lighter the color, the more stable the solutions were. And what you see on the left side in the full model is that starch biosynthesis destabilizes the metabolic system, while sucrose biosynthesis clearly stabilizes. And secondary metabolism has no real huge impact. However, if you miss photorespiration, then especially under conditions with high starch accumulation, secondary metabolism further stabilizes, supports the role of sucrose biosynthesis. And this gave us the idea that especially under challenging environmental conditions, when there's lots of starch accumulation, for example, during cold acclimation, then also the induction of secondary metabolism might support the stability of the CBBC. So finally, to conclude here on the role of the feedback inhibition of 2PG on metabolism, we further analyzed its role in stabilizing network by interacting and inhibiting TPI or SBPAs. And what we found was that TPI is heavily affected in its role in stabilizing the network. So the interaction of 2PG towards TPI gives a kind of an indication that this might serve as a regulatory switch. Some of you might ask, okay, what's new? That's what we know already, right? However, it is important for this model to make this difference because we now can, based on our eigenvalue spectra, which we calculated, predict, and that's good evidence coming from experiments, that there's a role and there's a meaning of the position of the TPI reaction within this full network compared to the SBPA's reaction. And we can discriminate by those two functions early reactions in the CBBC and regeneration reactions. And we further studied this in full regulatory background of the CBBC, which comprised about quite a number of simulations, and this was a challenge which was solved by my PhD Jacob Hernandez quite successfully. So in order to solve such a system, which includes all known interactions, regulatory interactions with a standard PC, this would have taken in our standard protocol 26 years, which is of course a bit too long. He managed to come up with an algorithm which reduces the calculation process for this to about 700 hours. And after 700 hours, what we found was that in context of the full up to now known and described by literature regulation of the CBBC, we still find that or can discriminate two functions here of 2PG inhibition on TPI and SBPAs. So the TPI inhibition seems to be a regulator for carbon allocation, while the SBPA stabilization or inhibition by 2PG has a kind of a stabilizing role for ribulose 1,5-bisphosphate regeneration. So to come to a summary of my talk, I hope that I could convince you that the structural and kinetic information provides some important information about plant-environment interactions. With such a model, we are able to predict regulatory sites and stabilizing sites, which might be also interesting for breeders, right? If you ask, how can I construct or breed a high yield crop, but which is still resistant towards temperature drops and so on, then you should account for such findings maybe, which tell us that the more stable you want to have the system, the less should come back in terms of carbon into the chloroplast. However, if you, of course, follow the natural pathway, then you will find that this is a kind of a trade-off function between biomass and stabilization. Further, we described that photorespiration stabilizes the CBBC to optimize, obviously, ratios of soluble carbohydrates and starch biosynthesis. And we found that secondary metabolism fine-tunes the stability of CBBC during acclimation procedures. So with this, I'm at the end of my talk. I thank you very much for listening. Would like to thank all our collaborators, funders, of course, my lab, and I'm happy to take questions.
Okay, so thank you very much. Please put your questions in the question box and they'll pop up here for me to ask Thomas. I'll start though. You talk about stabilization and this need for photorespiration for stabilization. So as a plant physiologist, how would I measure that? So let's say I've made a transgenic plant line, but I think this is going to overperform maybe by reducing photorespiration in some way. How do I then quantify stabilization? Is it like a whole crop season biomass, or what would I measure?
So our model, based on our model, we would measure the dynamics of metabolite concentrations after perturbation, and then quantify how fast, or if you come back to the same homeostasis as before perturbation, and if yes, how fast. So there are two possibilities at the beginning. Either you come back to the same homeostasis or you switch the homeostasis. I did not talk about the meaning of stabilization that much due to time issues. It might also be that certain instability is necessary in order to switch to a new metabolic homeostasis. So this is another trade-off. During acclimation, for example, we know that certain sugar amounts are brought to a stable new homeostasis, which we interpret as a transient instability, which allows the system to adjust to a new stable level. So here you would quantify metabolite amounts and maybe also enzyme activities, but it depends on the model system. If you model biomass allocation or biomass production, with specific interest in the export functions, then you might also quantify the biomass during or after such a stress.
Okay. I've got two questions that relate to the same thing from Thomas Altman. Which perturbations did you model, and if multiple, did you find the same effects?
Very, very interesting. Good question. So what we did is we perturbed the input function. So we simulated fluctuation of the carbon fixation rate, and we also perturbed the export function. And with regard to the differential impact, we can say that this was kind of indicated by the TPI reaction, that this sucrose export perturbation had a very strong impact on our photorespiration lacking model. So here this export function was kind of the dominating stability dominating function. I did not have time to go into detail. However, so what I can say is that you can correlate the reactions and the inhibitions which we simulate to this. And what we saw was that all interactions around the gap biosynthesis and interconversions mattered most.
Okay. So in terms of crop performance, if I was to try and, in terms of crop performance by increasing photosynthetic efficiency, you would want to keep photorespiration and maybe go for an alternative pathway to rather than actually suppressing photorespiration. So exactly. Is there an optimal amount of photorespiration in that case? Is it, and would it vary between species, and would it help us if we increase, say, SVPAs to improve photosynthetic efficiency, to also increase the amount of photorespiration even?
So what I can, it's hardly possible at the moment. I'm sorry for that. I cannot give you a clear number to that. What I can say is that due to our findings, we would come up with the solution which. So there are lots of bypass studies already, many of them also within the chloroplast. What we see is that obviously in our model, what would be optimal in terms of stabilizing the system and increasing yield is exporting, so starting the recovery of 2PG by export out of the chloroplast, but a fixation outside the chloroplast. So this is so not re-entering the plastidial metabolism anymore. So this is a kind of a clear message which comes from our stability analysis. So that's the only rough, or that's the most general aspect which we found. No.
And does it take into account any sort of C4 photosynthesis, any modelling on C4 photosynthesis where there is a suppression of photorespiration? Does that?
Yeah. No. So a clear no to this, because our model. So the approach is a structural kinetic one, and as soon as we change the structure of the network, at least we are not yet able to predict how the structural change would change our findings about stability. Yeah. So that's hardly predictable, at least for us at the moment. No.
Okay. Well, if there's any more questions, I think we maybe will come back to them at the end of the webinar for both speakers. And I think for now we will move on to our second speaker, Professor Erik Murchie. Okay, so Professor Erik Murchie holds a chair in applied plant physiology at the University of Nottingham, and his interests are in photosynthesis and plant physiology, focusing on metabolic and physiological responses to the environment, with a particular interest for crops under field conditions. His recent work of his group has included 3D architecture of cereal canopies, and the interaction with photosynthesis, manipulation of photoprotection in rice, nocturnal conductance in wheat, and exploring the diverse wheat and rice germplasm for dynamic photosynthesis and heat tolerance. And today he's going to talk to us about the challenges of moving photosynthesis between the lab and the field.
Okay, so finally, thank you for the chance to present to you. I think having heard Thomas speak, I think this talk fits in really nicely. After that, I decided to talk about canopy architecture. And I think that most of the work I'm presenting is part of collaborations with other groups, including CIMMYT in Mexico and the Vicky Plant Science Centre in Finland and others. I need to highlight Alexandra Burgess, who is a Lieberhuhn Fellow at Nottingham, soon to be Assistant Professor, and Jonathan Gibbs, a postdoc on the 4D project. So recently, I've been starting talks with this slide. We published a review with the word photosynthome with the intention that it describes all the processes that affect photosynthesis and its interaction with the environment. It's a bit of a cheesy name, but it does a good job of making the point that photosynthetic physiology is not a simple linear equation, but rather a complex number of interacting processes, including things like carboxylation, photoprotection, dynamic responses, the kind of things that Thomas was talking about just now. All of these interact, but it's canopy structure I'd like to focus on. So this meta-analysis came out a few years ago. I really love this term, pampered inside, pestered outside. It describes perfectly what happens when you try and compare plants in a growth room with those in the real world. You can replicate real world conditions and get meaningful results, but largely whenever you put a plant outside, it is pestered by changes in light, temperature, humidity, etc., and literally pestered. But it's a perfect description to me, and something of a relief, because somebody was considering how do you compare lab-grown plants with field-grown. And this meta-analysis came out with a number of conclusions, including the ratio of temperature to integrated light is important, and also things like planting density and of course pots. But the field should always be the goal. So I want to talk about canopy structure and microclimate and what's the role of architecture and movement in defining photosynthesis. So if you measure photosynthesis using an infrared gas analyzer or something similar in the field, and you measure a light response curve, you might get something like this. So that the red line would be a classic response of a leaf under good conditions. But high light, adverse temperatures, humidity, etc., lead to a down regulation, a pushing down of this curve. So, for example, under light saturated conditions, you have rubisco deactivation, particularly under high temperatures, stomodal conductance. Under the quantum yield light limited phase, you might have MPQ recovery and photoemission itself affecting CO2 assimilation. The point being that if you make a measurement of CO2 assimilation in the field, it may be less than optimal. And light fluctuations themselves cause this by slow induction and slow recovery from the highlight phases. So this is a crude cartoon of a canopy, and it models light in the canopy, and it's intended to show that when the sun moves overhead, you get a shifting of light caused by shading. So a mosaic of light patterning occurs throughout the canopy, and you can see this shown by the scaling of light. It's quite crude, but it shows it quite well. So on the right hand side, it shows data from a wheat canopy, where the 3D structure of the canopy was obtained by RGB imaging and reconstruction, followed by ray tracing. So ray tracing is a method for calculating the position of light in a 3D environment, such as a canopy from physical principles. And you can do this using multiple positions. So the direction of the source of light, I mean. So when you do this, you find a lot of light at the top, but moving down, you get bursts of light, these sunflecks that typically last minutes. And at the base of a dense canopy, it's common to find quite a low level of light, but sometimes just above the light compensation point, but punctuated with bright bursts. And it's the efficiency of photosynthesis tracking these bursts, which is causing a lot of interest at the moment. And so, but it's absolutely clear that canopy architecture influences yield. So recent empirical data showed that grain yield can be linked to the erectness of leaves. Simply placing leaves into an upright position allows better radiation penetration. So this is data from Richard Richards using a number of wheat lines, Australian wheat lines, and with varying erectness of leaves, and the upright leaves had higher high yield and higher biomass. And this is expected from earlier work on modeling of light. On the right hand side, you can see an upright canopy versus a flat leaf canopy, and the better penetration of light means that you get a higher photosynthetic rate, but less saturated than the canopy with the flat leaves. Okay, so it seems that we can use architecture to improve canopy photosynthesis, and there's a number of things to think about here. So not just the architecture of the canopy itself, but also things like the distribution of pigment composition, chlorophyll, and antenna size. And of course, biochemistry interacts with this in ways we don't fully understand, as I'll come on to. But I want to talk about movement, which is something that's less considered, but possibly very important. So it's the lower parts of the canopy that are, in our opinion, somewhat neglected because they receive substantial amounts of light. This figure on the left shows two wheat canopies differing in architecture, a tall upright and a shorter, more floppy structure. The lower intensities by the lower leaves, say below about 300 micromoles, make up a substantial percentage of the total light received by the whole canopy. So if the angle is suboptimal, in other words, the flat canopy, there's a whole lot more in blue at the bottom of the canopy here shown in the figure, and less at the higher light intensities. So in terms of ray tracing, this is ray tracing over a 12-hour period, so it's integrated light over the daytime. And so the efficiency of photosynthesis at lower light is clearly important. The figure on the right is a study where the leaves at the top were displaced at a frequency that's the same as a sort of light breeze, so about 6 meters per second. The movement of leaves creates more opportunities for light penetration, as we'll see. This is shown in red on the graphs here. Okay, we'll come back to that. Okay, so when we look at the literature, we find that most measurements of photosynthesis have been made at the top layer of canopies. This is pretty much convenience, but it's also thought that the flag leaf, in the case of cereals, the flag leaf is one of the most important contributors to yield. So to investigate this, one of our PhD students and now working in CIMMYT, Carlos Robles Zazueta, put this to the test using a combination of gas exchange, spectral reflectance, leaf area, and yield components analysis. And by calculating the photosynthesis ongoing in each layer, he was able to show that the lower canopy at pre-anthesis gave a better relationship with yield than the topmost layer. So this was less so for grain filling. So looking at these figures, this plots ASAT. So this is actually the saturated rate of photosynthesis, but integrated over the each of the layers, top, middle, and bottom, and plotted against yield. And we get a really nice correlation in the lower and middle parts of the canopy, actually much better than the top layer. And at 7 days after anthesis, during grain filling or Posada grain filling, this is less strong, partially understandable because at this stage the wheat plant is more sink limited. So, I realized that the function of lower layers in terms of contributing to yield is not new. This has been going on for a number of decades, but I think it has to be said that progress has not been good for understanding the physiology of these lower leaves, particularly in terms of photosynthesis, except in the case of improving responses of photosynthesis to fluctuating light that has been shown recently. And here we see a similar pattern as the previous one, but for radiation use efficiency, which is the ratio between biomass produced and radiation intercepted. Okay, so going back to the sake of time, I'll come back to wind. So in terms of wind, there are known effects on plants, things like development, heat and mass transfer, boundary layer conductance, microclimate, etc. And you can take a whole range of wind speeds on the Beaufort scale from 1 through to 12, that's sort of dead calm through to complete destruction. But in terms of this talk, I'm talking about light breeze. So these are not winds that cause damage, but rather winds that result in a minor movement of the canopy. So there's precious little on wind and photosynthesis, actually, if you look through the literature. This is a well-known study by Bob Piercy and Roden. So some tree species exhibit this thing called leaf flutter, which is caused by something called torsional gallop, which is a great name, where the petiole creates a self-perpetuating semi-rotation of the leaf in trees like aspen. Okay, this helps to improve light penetration to leaves below. So the leaves twisting in this way allows more light through. And they showed back in the early 90s that this enhances boundary layer conductance and also activates rubisco more than it would normally. And we published a paper a couple of years ago which shows that you can have a similar kind of effect for cereals. We call it isolated leaf movement, where the movement of leaves at the top creates opportunities for light penetration below. So the question is, what can we do about this? What is the variation and what effect does it have? So one problem actually is the definition of a sunfleck, because these light flecks caused by wind are extremely fast. So light can vary in quite complex ways, but a sunfleck would seem to need a beginning when it's low, a middle when it's high, and an end when it's low again. So this method by Maxime Durand and Matt Robson and Alex Burgess as well used a mathematical approach to identify thresholds to filter out background noise and identify sunfleck properties in time and space. So sunflecks are notoriously hard to define, but this paper devised a method of tracking light fluctuations using complex data that provides a repeatable way of defining them as brief periods of high light relative to the background. Okay. And by using a fast spectroradiometer, they also found that in fact the wind-induced sunflecks were more important, contributed more of the irradiance compared to the traditional sunflecks from solar movement. In other words, fast fluctuations are important. So working with the same Helsinki group, we compared two wheat cultivars, a tall curly-leaved canopy and a short upright canopy. The tall canopy Paragon also had a higher frequency of leaf movement compared to the short upright canopy. And if we look at just a static conformation here, so this is just the movement by the sun rather than wind as well. There were large differences in the duration and intensity of sunflecks between the two lines, but they were more intense in the shorter line, but shorter lasting at any canopy position. So architecture alone does determine duration and intensity of sunflecks, and it seems to be linked to leaf angle, with upright leaves getting, yes, better penetration, but also a longer time in highlight compared to lower layers. And if we look at dynamic canopies, these are the canopies in the wind measured using a fast spectroradiometer. We also found differences with the upright taller canopy having lower intensity wind flecks. Wind flecks is the word we use to distinguish wind-induced movement to sun movement-induced movement. So the wind flecks were longer, but they also had longer time between the wind flecks, up to 2 seconds. And this is a very recent paper from Maxime and Matt Robson. And I just want to highlight it because it showed, when you compare trees and crops, that the varied architecture, even within those groups and also between those groups, determines how much light comes from sunflex and windflex. Trees are very different. They have higher numbers of lignified stems, which gives something of a higher resistance. So you tend to get a more greater amplitude of movement. But even among the arable crops, the intensity and duration varied in a way that was also related to leaf angle and height. So here showing, for example, that leaf angle positively correlates with intensity and negatively with the clustering of windflex, the time between windflex. So, okay, so we get this variation in the patterning of light. So what about photosynthesis? I'm going back to this paper we published from 2016. We modeled CO2 assimilation in the canopy over the whole day in this canopy that was distorted, and so we simulated wind by displacement of the canopy. When we did this, we saw greater penetration of light, but particularly at midday. And when you model photosynthesis in the canopy in this way, you see up to about a 17% increase in daily photosynthesis when you move the canopy compared to when you don't move the canopy. And we've started measuring photosynthesis in replicating windflex. So this is a wheat leaf, and we replicate using an LED shifting with a 300 millisecond frequency between 100 and 1,000 micromoles. And here, going from 100 to constant 100 to this windflex situation, you do get a fairly rapid induction of photosynthesis. So this is, I guess, this is kind of expected given the dynamics of the Kelvin cycle and the electron transport chain, but it does show that these kinds of changes can induce photosynthesis. And down in the bottom right here, we see that when you do go through this, you increase the speed of induction of photosynthesis in the leaf. So the penetration of these very, very rapid flecks of light should have an effect on the speed of induction. Just a point that although these windflex are extremely rapid, so here we see data for an hour. This is taken in a wheat canopy, about halfway down a wheat canopy, in a period of wind. You can see these rapid sub-second changes in light. This is just a sample of that kind of data. That's taken in bright light. If you do the same thing in when it's overcast, you don't get the same kind of effect. So the speed of the number of fluctuations are less. So these kinds of fluctuations, whether they're sunflex or windflex, are really only occur when you have bright light at the top of the canopy. Okay, so what are we doing? We've been developing methods for modeling canopy movement. So this is tracking of a canopy, a wheat canopy movement using deep learning. This is Jonathan Gibbs, Alex Burgess, and others. And by tracking the movement of wheat ears in a canopy, identification and tracking of the ears, you can then follow their movement in a two-dimensional video, quantify the movement, and for the first time get data on the amplitude and frequency of movement of parts of the canopy. However, leaves are more difficult, partly because of the complex nature of the field, the amount of occlusion that occurs in the canopy. So we've been looking at new ways of trying to monitor and track and quantify movement in the field. Okay, and just briefly, this was work which is trying to model movement in 3 dimensions in the field and also the lab. This is work that again is ongoing by creating a box in the field with many video cameras. You can actually get a reasonable 4-dimensional model. It doesn't show particularly well here, but it's a reasonable 4-dimensional model of a plant moving in a windy environment. And we've linked this to a mathematical description for movement of a cereal leaf, which is developed by mathematicians on our 4D project at Nottingham. That's ongoing. Okay. And the last slide. So this is where we're at. What we really want to do is to go to the field and screen contrasting lines for movement properties in a high throughput way over long time periods, so we can get a reasonable idea of how this links to photosynthesis in the field. So this is in terms of active data collection, we're collaborating with Pietro Cicuta at Cambridge. And he's been using 2D methods to model cilia movement. In other words, the hairs in your lungs that pop up debris, etc. So he was using this method to image and model movement in those at that scale. But we got together and talked and thought, well, actually, this looks a lot like a canopy. When you think of cilia, it moves like a canopy and looks like a canopy. So we're developing his method together with him to do that. And this year we're combining physiology and microclimate data in the field with camera data and trying to apply this method to understand the impacts and mechanism of canopy movement on photosynthesis in the field. And I'll present this at the SCB meeting in Edinburgh. Okay, so what do we need to do? I think we need to understand how light fluctuation patterns become optimal with both canopy architecture and biochemical constraints, exactly the kinds of things that Thomas was talking about. We need to screen germplasm for movement and mechanical properties and understand the genetic basis and the implications of wind flex. Okay, I'll stop there. I thank all the people involved in this work. For the sake of time, I won't do it individually.
Okay.
Thank you.
Thank you very much. I have a question here already, so I'll kick off with this one from Arthur Nosalwicks. Apologies if I pronounced these names wrongly. Crops under fluctuating light are quite complex systems because of the plant structure with overlapping leaves, lower, older leaves of different spectral characteristics that are driven by different light in many aspects from direct sunlight. Is there a potential for improvement in lower or older leaves in regard to overall crop productivity?
Yes, that's an excellent question. Yeah, I think that because we get this relationship, the strong relationship between photosynthesis in lower leaves and yield, which was shown in Carlos's data, there's got to be options for this. So part of it relates to the work on dynamics of rubisco activation, MPQ recovery, this kind of thing, recovery from photoinhibition. This is, of course, highly relevant. I think the key is to optimize the sequence, the patterns of light, the frequency and duration of light, alongside those biochemical improvements. And the thing about senescence, I think, is correct, because there's this program of senescence and remobilization of nutrients going on at the same time. And at the same time, these leaves are acting as stores, but they're also acting as suppliers of carbon and also nitrogen for remobilization to the grain. So this pattern, the developmental pattern, needs to be taken into account too. I realize that answer is raises more questions than provides answers, but I think that's the approach we need to take.
So, in still conditions where there isn't any wind, promoting a planting potentially on a windy location is better in these terms, you would agree? But in that case, wouldn't you have to take into account root architecture, because you're going to have to start improving your root system and therefore needing better nutrient or use efficiency potentially, or it's going to use more nutrients for bigger roots?
There are implications for lodging. So what we don't fully understand is whether the movement needs to be entirely down to the petiole on the leaf, or how much stem movement we need. The issue with cereal canopies is that they're very densely planted and they tend to move together, and this may be a problem because it may limit movement. So it may be that we need more movement of both stems and leaves, or a more slower movement of leaves. The moving together thing with cereal canopies, which is a result of them being dense and also small, may limit light penetration. So the dwarfing of cereals may have actually limited light penetration as a result of wind. So the question then is whether we want to sort of loosen up the stems more, or whether we loosen up the leaves more. Whether we do the former, whether we do actually need to think about whether they are less or more likely to lodge. So yeah, absolutely.
I've got another question here from Teresa Fulga. Have you looked at the effects of wind flex on leaf temperature? And how would you say such fast changes affect leaf temperature, or is it too fast maybe?
Yes, so well, there's two things. The wind will reduce the boundary layer conductance, which may increase transpiration, which would cool the leaves down. This may be useful in a high light, high temperature environment where you have enough water. But you also got to be careful of increasing transpiration, and you reduce water use efficiency. There's some early work showing that if under very low wind speeds where you do get canopy movement, this effect is small. So the boundary layer conductance change is actually quite small under low wind speeds. But this is something that we do need to look at. We're kind of hoping that it is small and we don't have to think about it too much. But we will be measuring it.
Okay, I have, I think you may have answered this, but I'm going to just ask you the question. You consider the effect of plant architecture on photosynthesis, but there is the energy balance aspect especially related to conductance and water use, and also more light penetration can possibly affect leaf temperature. So is this something within your target goals, or have you considered it anyway? Maybe
yeah, similar to the last question. Yeah. It is the in terms of the microclimate within the canopy, you have a carbon dioxide drawdown quite often. So when you move the canopy and increase wind movement, you may actually help by introducing more CO2 into the canopy microclimate and negating a little bit the CO2 drawdown. But there is this potential problem of water loss under the low wind speeds that we're considering, which is 6 meters per second or lower. You probably will change the boundary layer conductance, but it shouldn't affect photosynthesis. You may get a higher transpiration rate, but again, I think we have to look at this in different environments. I think that the higher the temperature, the worse that problem might end up being. But it could be a trade-off. You're right. Yeah.
So just following on from that again from Mogus Retta, is the influence of leaf movement on the airflow significant, and would that affect your modelling?
On the airflow, I'm not sure whether that means the formation of these sort of turbulence across the top of the canopy. So cereals have a number of types of canopy movement. One of which is just the flickering of leaves, which is you get across the whole field. Then you get these waves of you see them as sort of waves going across the canopy, which are more complex to model and measure. But the 2D method that Pietro Secuta's group are using will actually allow us to look at those, which is one reason why that method's so attractive. The question is, I think the answer is I don't know. I'd love to know, and we'll try and find out.
Okay. If there's any further questions, put them in now, and if not, we're coming pretty close to the end of the webinar. So I think it's time for me now to thank our speakers. I'll just get rid of this. To thank our speakers for two really stimulating talks, and to remind you all that the next webinar is not until October, because we stop now for the summer, so all you people can go out there in the field and you'll be busy collecting data, no doubt. Our next webinar in October is with Dr. Pallavi Singh from the University of Essex, and Dr. Joanna Amaral from Lancaster University, talking about C4 regulation and rubisco activase regulation. If you have any exciting breakthroughs or photosynthetic research that you'd like to present in these webinars, this is my email address on the top of the screen, louisa.diva@seratium.eu. Give me an email and we can talk about whether you may want to do a webinar for us. But yes, that's all I have to say for you today. Great thanks to our speakers. Without the speakers and without the audience, these wouldn't happen. So I really appreciate you all turning up and your speakers for presenting for us. Thank you again. Until October. Bye-bye.